Bonjour, good morning, everyone. This morning we have the pleasure and the privilege to welcome Dr. Dan Roden. Dr. Roden was born and raised in Montreal, so that's probably the main reason why he's here today. But for us, he's a specialist in personalized medicine for quite a long time. From Montreal, he completed this medical degree and training in internal medicine at the Royal Vic, and then he moved to Vanderbilt University for an internship. It seems that his internship continues until now. At Vanderbilt, he directs the Vanderbilt DNA Data Bank Bioview. It's a discovery resource that, as of fall 2015, included over 200,010 samples linked to electronic medical records. Dr. Vanderbilt is also a leader in Vanderbilt Predict project that has, since 2010, has been embedded pharmacogenomic variant data in the electronic medical record of over 14,000 patients. He currently serves on the Advisory Council of the National Human Genome Research Institute. He has been elected to membership in the American Society for Clinical Investigation, the Association of American Physicians, and fellowship in the American Association for the Advancement of Science. Without further ado, I will invite Dr. Roden to present his talk, which the title is Tools for Personalizing Medicines from Patients to Populations. You got the pointer here? If you need it. Thank you.
Merci. Thank you. This is not, this is my hometown, but it's not the only reason that I'm here. One other reason is that when I talk in Montreal, it gives me an opportunity to use these funny two letters after MD. MD-CM is the degree that McGill gives and no one else gives. So I never show it because it just confuses people, but here I can show it. So that's an extra added advantage. So this is what I'm going to talk about. I'm not even going to read this slide to you, but I'm going to start with some pharmacogenetics stuff. So I'm going to talk a little bit about discovery, talk a little bit about populations, and then come back to the individual subject. So I came to Vanderbilt to be a clinical pharmacologist, and clinical pharmacologists study variability in drug action and mechanisms underlying variability in drug action. So if you expose a population of patients to a drug, good things happen to some of them in green, bad things happen to some of them in pink, terrible things happen to some of them in red, and nothing much happens to some of them in white. I'm fond of saying the commonest adverse drug event is the failure of the drug to do what it's supposed to do. So that's the people in white. And we like to segregate them, and everybody has seen slides like this. So the question that some people ask is why does this happen in a population? But if you're the patient, what you want to know is which color are you going to be? Are you going to be a green person or a red person or something in the middle? And there are many, many mechanisms that mediate whether you're going to be one or the other. So the history of pharmacogenetics starts with studies in very, very small numbers of subjects. This is a study that my mentor, Ray Woosley, did when he was a fellow in clinical pharmacology, moonlighting. So he was going to local coronary care units and looking at patients who were receiving procainamide. And one of the common adverse drug events during procainamide is development of anti-nuclear antibodies in the lupus syndrome. And he made the observation that was made by many other people previously, but in this particular way, he did it in a different and a better way. He showed that patients who were rapid acetylators for procainamide were protected against the lupus syndrome compared to slow acetylators. And the important thing about this study is that it involved 20 patients. So that was in the New England Journal of Medicine. When I came to Vanderbilt, we were studying a drug called enkinide, and we looked at its disposition as well as its pharmacodynamics, and we found that most patients, the ones in blue, responded to this drug in what we thought was a favorable way. I won't go into the details, and it may not have been such a favorable way in the end, but that's neither here nor there. But what the characteristic of the patients in blue is that they eliminated the drug quite rapidly, And they had terribly variable peak drug concentrations, 2 orders of magnitude, from 1 to 100 nanograms per ml. There was one non-responder that we studied, and she had extraordinarily high concentrations of drug and very, very slow elimination. That confused us for a while until we realized that enconite is a prodrug and required bioactivation, and it turned out the bioactivation pathway was CYP2D6. So this is an example of a drug that requires bioactivation to exert its pharmacologic effect, and that's the mechanism, major mechanism underlying variability in enconite action. And again, this study, also in the New England Journal of Medicine, involved 11 subjects. So small data as opposed to big data. And here is another example, also from Vanderbilt. I'm going to be very Vanderbilt-centric. I apologize in advance. This is a different polymorphism discovered by one of my mentors, Grant Wilkinson, who's the second to last author here. What he was doing is he was studying a drug called mefenitoin, and he gave mefenitoin to normal volunteers and then looked for presence and accumulation of the metabolite, the 4-hydroxy metabolite in urine. And you can see that there are normal metabolizers who accumulate the metabolite, and then there are slow metabolizers or poor metabolizers who fail to accumulate the metabolite. And to do this study, he actually required, he needed to study about 150 subjects. And so this is S-mefenitoin 4-hydroxylation, and I'll come back to S-mefenitoin 4-hydroxylation in a moment, because at the time I thought this was really pretty obscure. Nobody in this room has ever heard of or used the drug mefenitoin, and I thought he was wasting his time and everybody else's by describing this irrelevant polymorphism. So we'll come back to that in a moment. So now the vision in our world and in many parts of the world is that people will have sequence data and they will hand them to the healthcare system in some way, and the healthcare system will respond to that information in a rational and presumably cost-effective and healthcare-effective kind of way. This woman is handing her sequence data not to a physician but to a pharmacist. The pharmacist has no idea what to do with it. And the one thing I'm sure of is that when this happens, and I think it will happen, it will not be a piece of paper that will do this. It will be something else. When Francis Collins became director of the National Institutes of Health in the United States, he was interviewed by the New England Journal of Medicine, and he was asked what he thought of pharmacogenomics, and this is the exact sum total of what he had to say. You can read it, but I'm just going to highlight the fact that in the red box he said if everybody's DNA sequence is already in their electronic medical record, it is simply a matter of a click of a mouse to find out that sequence, then you can read down to the bottom. It should improve outcomes and reduce adverse events, and I agree with that vision completely, except for that one word, which I don't think is, and I can show you, and we'll talk a little bit about that. It is anything but simple. This is the medical record room at Vanderbilt University Hospital. In the mid-1990s, we were up to our eyeballs in manila folders like anybody else taking care of patients. And around this time is when we started to introduce an electronic record. I haven't held a manila folder in my hand for almost 20 years now. We started to use the electronic record seriously in about 2002. And these are the things that electronic health records can do for you, I think. One is to improve the care of individual patients. We can have a long debate about that, and I'm not going to even discuss that. But at the level of taking care of patients in clinic, I think it is a boon done right. We think we can use the information, if it's aggregated, to identify specific subsets of patients. How many types of diabetes are there, really? How many types of heart failure are there, really? We can use the platform as a platform for discovery. We can use it for drug development and drug repurposing. I'll talk a little bit about both of those. And then if the vision of the lady with the sequence handing it to the pharmacist is ever going to happen, it's going to happen in an electronic medical record environment. So the implementation of genomic medicine and pharmacogenomic medicine is not going to happen in a paper environment. It's going to happen in an electronic environment. So what we've done at Vanderbilt with I would say support, but I would say more pressure by Vanderbilt leadership, who really embraced this idea, is we've made a huge investment not only in clinical pharmacology, the sort of one underpinning discipline, but in biomedical informatics. So we have a very, very large department of biomedical informatics which runs our electronic health record. So the first thing we did was we took our electronic health record, 2.6 million records, and de-identified them and made them into an image, which I'll talk a little bit about in 1 second. We call it the image the synthetic derivative because over time this image of the electronic record should become a more useful tool for mining information than the actual electronic record itself. So the way we do this is we start with clinical notes, test reports, whatever is in the electronic health record. We have an integrated health record, all the same across inpatient and outpatient environments. A typical note would look something like this, and for those of you who were not medically oriented, T2D is type 2 diabetes, and RA means rheumatoid arthritis, but it could also mean rheumare, or it could also mean right atrium. So the system to look at this kind of information and to curate it has to be moderately sensitive to those kinds of nuances. So the first thing we do is we take out the identifiers because that's the way our data bank is constructed. The next thing we do is we take out medications and we structure the medications properly, and then we sieve the rest of the information and put it into a structured format. So you can see how the synthetic derivative, as this happens over time, and I would love to say we've done this with all the records and they're perfect, of course they're not. You can see how over time this becomes a much more interesting and useful resource for biomedical discovery than the original electronic record. The other thing we do is we develop custom queries. Some of the queries are, does this patient have a disease like type 2 diabetes or rheumatoid arthritis? Some of the queries are, is this person a smoker? Some of the queries might be, does this person live close to a grocery store? Because those are important determinants of health too. So as we do the queries, we run them, and we run them and we put them into the synthetic derivative as well. So over time, you can look at the record of an individual subject, and it becomes a more useful way of sieving and looking at the electronic record than the original electronic health record. A subset of our patients have DNA samples. The number is currently over 215,000. I'd love to say they've all been sequenced, and sadly they have not been. In fact, we have really a modest amount of genotyping and of genotyping information, mainly from GWAS, which is now about 25,000 subjects, and exome chip data from the first version of the exome chip. We're currently in the process of a very large upgrade of this data set. So we're doing about 5,000 to 10,000 subjects per month over the next year. So we're going to bring that number up to over 100,000. And my idea is to bring it all the way to 215,000, at least dense chip data. The entire synthetic derivative lives on a high-speed search engine called a Netezza. The beauty of this is that I can access it anywhere in the world. I can access this interface and ask the question, do we have patients with this disease or with this characteristic? So here is an example, and in the time it takes me to show these slides, that's how long it would take me to actually do the search. So I asked how many subjects have an exposure to clopidogrel, and the number is around 50,000. You can see it there. And then I can ask, and how many of those have coronary stenting procedures? And there's lots and lots of procedure codes for that, so I have to drag and drop each one of them, and we're fixing that. And then the intersection of that set is, as you can see, around 12,000. And then when I click on the little button that says, there's a little button down in the lower right-hand corner that says only samples with DNA, and of course there's a subset with DNA. So immediately I know that there's a set of something like 3,000 or 4,000 subjects with clopidogrel and a coronary stent. So I can do something with that data set. And I can click and find out how many of them are men, how many are women, how many are African American, what age distributions they are. And this all happens immediately. So using those kinds of tools, a curated, moderately curated phenome, and rapid access to summary data, and then actually to individual data, we've evolved this kind of approach to case control studies for risk across our biobank and across multiple other biobanks with which we are affiliated in something called the Electronic Medical Records and Genomics, or the eMERGE network that's NHGRI funded. So we define cases, we define controls, we develop algorithms for both of those. I can talk a long time about how those algorithms perform. or not, as the case may be. We sometimes identify possible cases which require manual review. Sometimes we use those, sometimes we don't. And then we can do case control studies. And what I can tell you since the Electronic Medical Records Genomics Network started in 2007 is that we can find algorithms, we can validate them, we can replicate them across multiple electronic health record systems. They perform pretty well. We can replicate any known association with common diseases like type 2 diabetes, atrial fibrillation, rheumatoid arthritis, what have you. When we have large numbers, we replicate that really, really easily. And the other thing we've discovered is that as you accumulate large sets of genomic data in patients with electronic health records, you can start to ask new questions that can be answered without the need to do any more genotyping. The genotypes are there. They were obtained for some other reason. They're sitting there, and we've done lots and lots of studies with existing genotyping and no extra charge to us or our funders. So in Bioview, we've had one focus on pharmacogenomic signals, and here's one. So we've asked the question, what is the risk for cardiovascular events after clopidogrel for coronary stenting as a function of S-mephenetoin hydroxylase metabolizer status? And I mentioned that enzyme earlier as sort of an irrelevant enzyme. Well, it turned out that S-mephenetoin hydroxylase, and it's hydroxylase, not hydrolase, is actually CYP2C19. So when Grant Wilkinson discovered this obscure gene working in an obscure laboratory in clinical pharmacology at Vanderbilt, he actually did the much larger pharmacogenomics community a big service. So if you look at prospectively obtained data looked at retrospectively, these are data from Jess Mega, but there are many others. If you are a carrier of one of the variants, one of the loss-of-function variants in CYP2C19, you don't bioactivate clopidogrel. Clopidogrel is like enconide, it's a prodrug. And therefore, you don't have active antiplatelet effects, and you have a 12% incidence of major adverse coronary events over the course of a year. If you do not carry one of those loss-of-function variants, the incidence is 8%. We looked at our electronic record, and we find really very much the same kind of outcome with 807 patients at the time, and there's lots more now. We've used our electronic health record as a test bed for pharmacogenomic discovery, so that was a validation study, but we've done, these are all the phenotypes that we've looked at, the ones, and the interesting thing is, again, these data can be reused, so a patient who is a case in one study can be a control in another if they receive the other drug. And these are the ones that have been reported, most of them in full-length publications. And here are 4 examples of new discovery for drug response phenotypes using genome-wide association. And of course, genome-wide association is the first step to understanding the mechanisms underlying these kinds of variability, and we're working on all of these still. Another thing that I'm very proud of, another story that I love to tell, is this story about NPC1L1. So NPC1L1 is the target for a drug called ezetimibe. It's a known cholesterol-lowering agent. There was some question about whether ezetimibe would actually protect against coronary disease. So Seket Theresin, a very distinguished lipid investigator at the Broad Institute, Massachusetts General Hospital, did a study where he looked for loss-of-function variants in NPC1L1, so variants that would mimic the pharmacologic effect, and found them and found that they protected against coronary disease. Had a set of 53,000 subjects, sent it to the New England Journal of Medicine. They said, that's very interesting, we want you to get more subjects. So I got an email on a Saturday morning from him saying, do you guys have subjects with coronary disease and this particular variant? So it turns out the variant he was interested in was on the exome chips, which we had already run. We had validated algorithms for patients with coronary disease and controls for patients who didn't. So within a day, I could give him a large data set. It took about 2 weeks to produce the final data set because we had to find all the covariates like smoking and diabetes and hypertension. But the paper was eventually published with 113,000 subjects, 30,000 cases, the rest controls, and you can see that there is a protective effect of these very, very, very rare alleles. And the most important thing is that the largest contributor to this data set was Bioview. So once the data is in place, once the data are in place, you can do these kinds of studies pretty expeditiously and contribute in a big way to greater discovery. So I've already told you that we have curated the phenome. What else can you do with a curated phenome? Well, in general, a typical genetic experiment starts with a phenotype. failure of clopidogrel to prevent major adverse coronary events, susceptibility to breast cancer. You would name the phenotype, it's there. Baldness, macular degeneration, what have you. So, and then tries to identify associated genotypes using a variety of approaches. So one typical approach is a genome-wide association study, and the data are typically displayed this way, as you know. So we said, well, what happens if we turn the experiment on its head? And if we have a large number of subjects who have genotype data and who have lots and lots of phenotype data that's been curated, then you should be able to do something like this. You start with a genotype and ask the question, if somebody is reference or variant at a particular genotype, with what phenotype across the electronic medical record does that associate? And so we've done a lot of different kinds of experiments using this approach. And these are some examples. I can't, if I had an hour or two, I could talk about lots more of these. But we've done a lot of replication, and I'm bored with replication because what we replicate is real, and what we don't replicate we sometimes think is a GWAS signal that's probably not real. I'll talk a little bit about pleiotropy, and I'll tell you a little bit about drug repurposing, and then there's some really interesting ways of engaging the basic science community that I'll give you an example of that. So here is an example of pleiotropy. I would love to say it's a typical example. It's, of course, our best example. This is what happens when you do phenome scanning with a variant in IRF4 that's known to be associated with hair and eye color. And you can see there's a striking association with actinic keratosis with a p-value that's 10 to the minus 20 something or other. And then there are associations with various kinds of non-melanoma skin cancers. There's also an association, now I have to use the pointer, because people won't see this usually. I'm going to point over here. There's an association with something called solar dermatitis. So solar dermatitis is just a fancy way of saying sunburn. So this is the world's most complicated way of saying that people with red hair are sensitive to sunburns. And that's what we discovered. There's also an osteopenia signal, and that might be a false positive, but it might have something to do with sun exposure and vitamin D. So we think that these are interesting associations. This is one of my current favorite stories. A friend of mine who I've known for a long, long time, who is an expert in how impulse propagation works in the mouse heart. There's a specialized conduction system in the mouse heart, and there's a set of genes that he's defined in the conduction system. So he has one gene that's one of his favorites. He sends, he's knocked it out, and he's shown how important it is in the mouse heart, sends it to one of the very, very high impact journals, and their response is, that's really interesting, but what does this have to do with humans? And he emails me and says, can you help with your magic? And we said, well, sure. And we looked, and I can't tell you the name of the gene, of course, but this is what the phenome scan looks like in an Excel spreadsheet. So this is the way we usually get it before we display the data. And what's really striking about this one is that the top associations are with conduction system diseases. So this kind of result, you'd sort of say, well, okay, that's pretty interesting. The p-value is 10 to the minus 4th, 10 to the minus 5th, which is borderline for this kind of analysis, we think. We argue a lot about where the p-values should be. And the mouse stuff is interesting, but taken together, they really make a very compelling story. And I think that's one of the lessons of personalized medicine as we go forward. Data from multiple types of data sets are going to be important to validate these signals, make sense of them, and actually figure out what it is to take forward into patients. We've done phenome scanning on known drug targets. This is a list of drugs and their known targets. We scan for variants in those genes and then ask the question with what phenotypes are those variants associated, and what happens is that we recover the disease for which the drug is prescribed. And that's a very sort of reassuring result, because one of the things that we have in our sites, and we have data that this works, is taking drugs that are known. So here's an example of a drug that's on the market and it has a known target. I just have to emphasize that right now these are made up until we publish the real data. I have to make up these names. And let's pretend that it's indicated for rheumatoid or psoriatic arthritis. So what we do is we take SNPs in the target gene and we ask which one of those SNPs are associated with rheumatoid arthritis or psoriatic arthritis. And those are the ones that we think are real or functional. and we take them forward and we scan the rest of the phenome to look for new indications and also for potential adverse drug events during therapy with this drug arthriticide. So Francis Collins had this idea, and we share this idea of sticking pharmacogenomic information into the electronic health record. So we created a project called PREDICT, and this is what PREDICT stands for. I can never remember, so I have to put it on the slide. And the idea in PREDICT was to find, so let me back up, and let me just say that if we believe in the idea that everybody in this room is going to have genomic variation stuck into their electronic record, what is the first step you would take? You're certainly not going to start to sequence everybody in this room and stick their data into the chart. You have to figure out who you're going to start with and what drugs you're going to start with and what variants you're going to start with. So we decided to select patients who we thought were at high risk for receiving a drug that has a pharmacogenomic story. So for example, at the time we were starting this, patients who were getting elective knee or hip replacement were routinely put on warfarin afterwards. Still are in some environments. So that would be a population at high risk. You identify them ahead of time. Patients who are going to the coronary to the cardiac catheterization laboratory were at high risk for getting clopidogrel because a lot of them get stents, and after stents they get clopidogrel. So we would take those high-risk patients, genotype them, not just for the variant that we thought might be important for the drug we thought they might get, but also for a lot of other variants for other drugs that have pharmacogenomic stories. We archive all that information. We reveal some of it in the electronic record once we're sure that the signals are right and once we know what to do with the information that we're going to present to doctors. So as we're planning this study, the FDA puts a black box around clopidogrel, and you can read the black box if you want to. The bottom bullet is the most important. Consider alternate treatment or treatment strategies in patients identified as CYP2C19 poor metabolizers. So that doesn't say to genotype them, but it says if you happen to know, then you should consider alternate treatments. And I can't resist this editorial comment. Everybody wants us to provide data that are black and white. If you are a poor metabolizer for clopidogrel, you will die in the next week. If you're a rapid metabolizer for clopidogrel, you will live forever. And that's what people want. But of course, what genomic data does, and pharmacogenomic data in particular often does, is that it just shades the odds of having a particular desired or undesirable response. So just as an example, these are the source data for clopidogrel. An investigator in France, Jean-Sébastien Hulot, did this study in 2006. Clopidogrel got on the market in 1998 in the United States. It's known to be a prodrug. The pathway for bioactivation was not defined. So after a bunch of in vitro studies, he did a study where he took a group of patients who were known to be heterozygotes for the loss of function variant, the common loss of function variant STAR2. So that's the group in the white dots, and he compared their responses to clopidogrel to a group of patients who were known not to carry that variant, the STAR1 group. So at baseline, the platelet function, which is shown on the y-axis, was the same in the two groups. And then he gave them 75 milligrams of clopidogrel for a week. So that's the usual clinical dose. And you can see that there is an antiplatelet effect in the STAR1. So they bioactivate clopidogrel to its active metabolite or metabolites, and they have an antiplatelet effect. What you can also see is there is striking variability in that effect. So some patients have no antiplatelet effect, some patients have a huge antiplatelet effect. And in the group that are the STAR1, STAR2s, on average, there is no antiplatelet effect, but there are certainly some who appear to have an antiplatelet effect. So this is the way pharmacogenomics works in the real world down at the level of the real single individual patient. So if on average you knew that somebody was a STAR2, you might consider alternate treatment. But you wouldn't on average, and that's the best we can do right now. What's not included here are patients who are homozygotes for STAR2. I'll show you a little bit of data on them. They're about 2% of the population, and they really are predicted to not respond to clopidogrel. There are data that if you're a heterozygote, you'll respond to a higher dose, but if you're a homozygote, it doesn't matter how much you give, you won't bioactivate the drug and you won't get an antiplatelet effect. So that accounts for this effect that Jess Mega reported and that I already showed you. So it's a 12% versus 8%. It's not 0% versus 100%. So we've been doing PREDICT for a while now at Vanderbilt, and this is what the electronic health record looks like in a patient who has had PREDICT testing. You can see, again, I have to sort of point it over here. You can see right in this box here, we report the raw genomic data. Most physicians have no idea what to do with that. So the way the system works is if an electronic prescription for clopidogrel is written, should I smile? Is written, the system looks, if there's genetic data, the system looks further and says, is there variant genetic data? And if there's variant genetic data, then the system reports back this person has a variant, and this is in real time. This is what's so-called real-time clinical decision support. Reports back that that there's variant data and suggests alternate treatments, and allows the physician to continue the clopidogrel, but asks for reasons why. We've actually, and I didn't include these slides, we've actually reported in the last 6 months 2 studies on what happens. One is if patients have this kind of information, physicians switch to an alternate form of therapy about 60% of the time in the homozygotes and about 30% of the time in the heterozygotes. We're very interested in knowing why they switch rates aren't higher, but the fact is, and they switch about 5% of the time in the patients who are wild type. So there's a clear impact of the genetic prescribing information. It's not as big as you might think, but it's pretty big, particularly in the homozygotes, which is the big target population. The other thing we've started to look at is outcomes. There's a little bit of data reported in an abstract that this actually does improve outcomes in terms of major adverse coronary events, but we're still looking, and we're putting together a very large data set across multiple institutions that are doing this. So this is what happens when you look. There are 2.7% of the patients are homozygous and about 20% are heterozygous, and the rest don't have a variant. So if you're a cardiologist, you say, why am I doing this? I'm looking for the 2% of patients who have this variant. There's tremendous variability across the spectrum of response to clopidogrel anyway. It's probably drowned out by the genomics. So there's a big pushback to doing this, and I understand that. We've looked in our data set across the first 10,000 PREDICT subjects, and we asked this very simple question: how many patients have a variant that would be predicted to really confer a highly undesirable drug effect? So for clopidogrel, that's the homozygotes, and that's, in this particular slide, it's 2.5%. And then there's a group of people who are in blue who have a somewhat undesirable effect, and that's around 20% for clopidogrel. So we looked across 5 drug gene pairs. You can see them at the bottom, and just ask the question, how many patients are there altogether who have some variant that might modulate response? And the answer is that about 5% of patients have something that would produce a really undesirable effect if they got that target drug. And they may or may not ever get that target drug, but that's the way it is. And about 91% have something that they might know about. So that's just with 5 drug gene pairs. So we think that what this is telling us is that everybody has something. You don't know what it is that you have, but you have something that will modulate variability in a response to a drug. So this is an argument, I think, for multiplexing these data and doing them all at once, as opposed to the one-off, doing the clopidogrel test or doing the warfarin test or doing the TPMT test or whatever. So we do it in a multiplexed way now. One of the challenges is to educate the physician audience and the healthcare provider audience. So we've put up a Coursera course called Case Studies in Personalized Medicine. Here is me trying to explain sensitivity and specificity in one of those talks. That talk went live on January 15th. There have been about 5,000 participants so far, so you too can look at this course. And there's hours and hours of lectures about genomic medicine and pharmacogenomics and implementation there. You'll recognize some of the slides, in fact, if you go there. So another project that we've done in the eMERGE network, basically in the eMERGE network we asked the question, what happens if you replicate the PREDICT model at Vanderbilt? So, but instead of doing genotyping for specific target variants in these drug metabolizing genes, we used a platform that actually interrogates 82 key pharmacogenes. So those are drug metabolism genes, drug transporters, and drug targets. And so we're going to get lots and lots of variation, most of which we don't understand. And I'll come back to that in a moment. So the 5 or 10 or 15 actionable variants, each site set up a system like PREDICT where we have clinical decision support. And for the others, we just created a repository for people to look at and ask the question, are there unusual drug responses as a function of genotype? So one of the problems that we encountered, and I think is going to be an enormous problem going forward, is the so-called variant of uncertain significance problem. So on the platform, there are 2 genes, SCN5A and KCNH2, which are known ion channel genes in which mutations, rare variants, cause something called the congenital long QT syndrome. When I'm not being a clinical pharmacologist, and I'm not being a personalized medicine person, I'm a cardiologist who takes care of patients who have this disease. So this is sort of one of my other hobbies or specialties or whatever you want to call it. So because these 2 genes were on the platform, we went back and looked at how many variants we had. And it turns out of the first 2,000 people in eMERGE PGX, we've now done 9,000 people. So this is just the first 2,000. We had 127 non-synonymous variants. Almost all of those are rare, and in fact most of those are singletons. And as you sequence people, you find, every time you sequence, you find something new that's never been reported before. And then we sent those 121 variants to 3 separate laboratories that do this for a living, and asked them to assign pathogenicity. What would you do if you found this variant? So one lab said 16 of those are pathogenic. Another lab said 24 are pathogenic. Another lab said 17 are pathogenic. And they agreed on almost nothing. So there's an issue in establishing pathogenicity here. Then we're the Electronic Medical Records Consortium. So we went back into the electronic records. We found 1 patient with atrial fibrillation, which is at least the background rate. 2/3 of the patients had electrocardiograms, and almost none of them had a long QT syndrome. So this raises a whole bunch of questions. Do you tell patients that they have a variant of uncertain significance? Do you tell patients they should have an ECG? Do you screen the entire family because there's variable penetrance of these genes? Which family members do you screen? If you tell the patient they have something benign and it turns out you change your mind, how do you get hold of them to tell them that you've changed your mind? So this story is repeating itself over and over again. As we do genome sequencing, we find patients who, by what we know in their genomes, should be dead, and yet they're not. And so this is an example of the same thing, and we're going to struggle with this in the reporting world for a long time. So those are big data sets. I want to come back just for a second to the individual patient, because I think there's something to be said for that. So again, I told you I'm a cardiac arrhythmia person, and one of the phenotypes that I've studied for the last couple of decades is something called the drug-induced long QT syndrome. So this is a patient who has a normal electrocardiogram, except for a couple of premature atrial contractions. At least I think it's normal. I can't see it from here. Yes. And their QT interval, which is marked on the slide, is within normal limits and a little on the long side. They get 2 doses of sotalol, and they get spectacular QT prolongation. The QT interval should never be above 500 milliseconds. So this is really, really, really long. And the problem is not the electrocardiographic epiphenomenon. The problem is that once patients get long QT intervals, they're susceptible to doing this particular abnormal rhythm called torsade de pointe, and this can be fatal or at least cause syncope. So we've spent a lot of time trying to understand the genomics of this, and I won't bore you with that part of the story, but I'll just tell you that there are data, these are data from Germany, that if you have had torsade de pointe and you're re-challenged with a QT prolonging drug, you'll develop an exaggerated response. So it suggests there's a genomic basis, or there's some kind of difference in cardiac behavior on exposure to a drug. So what they did in this study was they took 22 patients who had had drug-induced torsade of the type I just showed you. They waited until their QT intervals came back to normal after the challenge with the drug. They brought them to the coronary care unit, and they gave them a challenge with intravenous sotalol, a QT prolonging drug. And you can see in the right-hand panel that everybody's QT interval gets very, very, very long. In a group of control subjects who did not have drug-induced torsade, the QT interval also gets long, but not much longer than at baseline. The other interesting thing is that the two groups are really pretty indistinguishable at baseline. It's only after drug challenge that the differences become obvious. So I don't think we can do that study in our coronary care unit, and I don't think, I dare say that it would be hard to do in any Western coronary care unit right now. So the question then we ask is, to what extent is the risk predictable in an individual subject? What we would like to do is to compare the biology and pharmacology of those cardiomyocytes from patients who are susceptible and patients who are not susceptible, both at baseline and after exposure to drugs that prolong the QT interval through known QT modulating pathways, which I won't bore you with. So cardiomyocytes is the key word here, and it's only in the last couple of years that we've managed to sort of get the technology to where we think we can do this study. So this is a study that we are now in the middle of. Take two subjects who have had one excellent response and one terrible response to a drug like sotalol, make induced pluripotent stem cells, drive those into the cardiac lineage, and then ask the question, what do they look like at baseline, and what do they look like after drug challenge? And you can see that we record action potentials, and action potentials is our surrogate for the QT interval. And then take those data back and to try to use predictive measures that we develop in vitro to identify patients at risk in a broad population. So that's an example of many, many kinds of examples which people are using these kinds of cellular technologies at the individual subject level to understand variation in the genome and how that affects drug response and other kinds of phenotypes. So in order to get to those individual subjects who have excellent responses or bad responses, you have to have a large population to draw from to identify those extremes of response. So there are a number of very, very large data sets that have been accrued around the world. UK Biobank is 500,000, the Kadoorie Biobank is 500,000. The Million Veterans Program in the United States is currently pushing on 500,000. They're hoping to pass the UK Biobank in the next couple of months. In the Emerge network right now, there are 400,000 subjects who have DNA samples and electronic health records, and about 60,000 of those have GWAS data that's been imputed. So it's a very, very large data set now. There's the DECODE set, and there are other data sets with which many of you are familiar. So the question is, why do you accumulate these very large data sets? And I call this the paradox of personalized medicine. In order to treat one patient differently from the average, you can't do an N of 1 experiment in general. You have to have a subset of patients who behave differently for some defined reason. So if you're going to study extreme drug responses, either great drug responses or terrible drug responses, or if you're going to study extreme genotypes like the NPC1L1 story, you have to have these very large data sets to draw from to make inferences on how to treat an individual subject. I also think that's going to be a great resource for enabling phenome scanning for discovery and lots of other applications which I won't talk about. So the last speaker ended with a quote from Osler, and I'm sure that you all know that Osler, before he went to Oxford, was at Hopkins, and before he was at Hopkins, he was at Penn, and before he was at Penn, he was at McGill. And the Osler papers are still at McGill. They're not at Penn or Hopkins or Oxford. But before there was Osler, there were other people who recognized that there is individual susceptibility to disease and drug responses, and starting with Hippocrates, I'm sure there are earlier examples. More important to know what sort of a patient has a disease than to know what sort of a disease a person has. So that idea has now been embraced in the United States by President Obama and the healthcare people around him in the Precision Medicine Initiative. And the Precision Medicine Initiative includes a number of components, one of which is to build a cohort of at least 1 million. They say 1 million, but I hope the operative word is at least 1 million U.S. Patients, with the idea of making this a recontactable data set. And the word we're not supposed to use when we describe this is patients. These are participants. They're fully engaged in their healthcare. They understand what the genome looks like. They understand what the variants are. And that's going to be a major challenge and lots of fun to try to execute. And the initial data sets are going to be electronic health record data, specific targeted questionnaires, biospecimens, and some data obtained through data streams through your smartphone. And we'll see what the smartphone data streams look like, but those are going to be an interesting data set. So the precision medicine implementation timeline looks like this. The project was announced in January 2015. It took from the announcement till the fall of 2015 to generate a blueprint for what the project might look like. The very first grants were awarded in the spring of 2016, and they were awarded jointly to us and to Google to start the process of designing this in a way that would enable the whole process to go forward. And the project is really going to start to sort of accelerate over the summer and the fall. And the target is to have about 80,000 subjects enrolled by early November, when something happens in the United States. I can't remember what it is. So this is my Osler slide. I don't show a picture of Osler. I just show a picture of my friend John McPherson, who's an interventional cardiologist, and his patient. And I'm fond of this picture because this is the first patient that we had in our PREDICT program who was a STAR2 CYP2C19 poor metabolizer. John had just put a stent into her, so aside from all the other things he knows about her personal life preferences, her personal life history, her other diseases, her other medications, he now knows a little bit more about her, and so he can personalize her care with respect to clopidogrel and with respect to all the other things that he does for her in a more rational way. So that's the goal of personalized medicine at one level, the individual patient, but it's going to take large data sets to get there. These are the teams that we've engaged to do that. There are lots and lots of people here, and I won't even, I'm not going to highlight any specific names. I'll just say that there's the Biomedical Informatics Brain Trust, there's the Ethics Brain Trust, there's the Genomics Brain Trust, there's the Pharmacogenomics Brain Trust. There's a bunch of fellows. And then the lower right-hand corner are the people who run the medical center who have embraced this vision at least as powerfully as I am. So again, I appreciate the opportunity to visit my hometown, and I'll be glad to take questions.
Thank you so much, Dr. Roden, for this very inspiring lecture. So, is there any questions?
Go.
Please go to the mic.
Dr. Roden, thank you very much for a terrific overview of personalized medicine and why it's important. My question, the answer to my question may be implicit in what you said, but I want to ask it directly. When we look at personalized medicine, we inevitably point to all the progress made in oncology and wish for more progress outside of that particular disease, especially in cardiology and CNS disorders. Can you reflect on why that's the case, that we've made less progress in the past 10, 15 years outside of oncology?
So I don't think I'd argue with the idea that that a huge amount of the progress has been on oncology. I obviously didn't touch on that because I'm not an oncologist and because the previous speaker did. The facetious answer is that I would love to be able to get to patients' tissue. And if I ask a patient for a piece of heart, the likelihood that they'll say, yes, it's small. It's not zero, but it's small. And if an oncologist asks a patient for a piece of their tumor, the answer is almost certainly, why not just take the whole thing and not a piece? So they have been able to exploit genomic technologies in that way. That's a facetious answer, though. I think the real answer is that it's pretty clear that cancer is a disease of the genome. And with the development of high-throughput, next-generation approaches to sequencing, that nut has finally begun to be cracked. You know, if you cast your mind back 20 years, people said, well, it probably has something to do with the genome. But now that you have real genetic variants that turn out to be targets in many cases, I think those are game changers. The extent to which heart disease is a genomic disease is less than in cancer, I suspect. But we have lots of questions we don't have answers to yet. So I'm hopeful that some of these new technologies, like the IPS technologies, which obviously can be used for lots of other things besides the example I showed, can start to sort of show us where the answers are. One thought that people have, or one criticism that people have, is that there are a couple of poster children for pharmacogenomic implementation in cardiology, and they're drugs that are going to go away, clopidogrel and warfarin, and that's fine. I think they've sort of taught us, mechanisms underlying variability, but the idea that now that those drugs are going to go away, every drug will have uniform and tight responses to therapy is far-fetched. I think that every drug will have a pharmacogenomic story, some of them bigger than others, some of them common variant, some of them rare variant. So I think we're just beginning to scratch the surface, but oncology is different because oncology is a disease of the genome largely. I think that's the explanation.
Thank you.
Thanks, Ed.
I have two questions. The first one is about the large number of people who have one of the variants that modifies the effect of drugs. You have one slide about that. Today, with about $30, you can run an array from Illumina that's with imputation analyze about 5-6 million of SNPs, which means that it would be extremely easy and inexpensive to test the entire population and to develop a very simple device that's these kind of things. Mm-hmm. And every physician can put that in a computer, and with a very simple software, they can type a drug and see if the patient has a mutation. That will cost about nothing, it will save life. Why it's not done? Well, first question.
So you sound like Francis Collins, because it's simple and it's
you.
easy
Thank you
and
very
it'll
much.
be done. Yeah, I share that vision. The problem comes down to things like, so the example I gave was clopidogrel, and in some ways it's the exception, because it relies on this idea, number one, of bioactivation, which is not a completely common phenomenon, and number two, on common variation. So we know what to do with clopidogrel because the variants are common and they've been studied. I can guarantee you that if you start to type people on an Illumina platform or anything else, first of all, you're going to miss some things unless you sequence. But the vast majority of stuff that you type, you won't know whether it's a loss of function or a gain of function or a nothing. So that was one of the reasons that we started this eMERGE PGX project, to start to get sequence data for these things, to see how common variants are, how rare they are, and actually whether we can associate any other variants with drug response phenotypes in the electronic health record. And that project actually continues in the current round of eMERGE, where instead of doing 2,000 subjects and 2 genes, we're doing 25,000 subjects in over 100 important genes. So I think the question is which variants are actionable, and then the business of building the clinical decision support systems relies on the idea that you actually have evidence for what to do with each of those, and the evidence is that you'll do something good as opposed to something bad. Occasionally you sort of worry that you sort of say, that person shouldn't get this drug, they should get that drug. They get that drug and they have an adverse event, and you say, well, maybe they should have got this drug instead. So you have to have the evidence as well. Well, so while it sounds simple, and I'm with you, I think that we're going to see large-scale typing, or sequencing, or something. There's a lot of things that have to get done between now and then.
I understand what you say. There's one question: is research is research, and practice is practice.
Right.
There are some SNPs that has been for sure shown to modulate the effect of drugs, and if we can give the advantage to the patient and giving them their genotype is something you can do today, not in 5 years.
We're doing it. We're doing it at
Yeah.
our place, and there are a handful of other places in the United
Yeah.
States that are doing it. I mean, but it's not like you're going to go from nothing to everything over the course of 6 months. It's going to take a little while until people adopt, people see the value, and we actually, it's up to us to actually show some of the value. And that's what
So
we're
my
trying to
second
do.
question is about the JAMA paper, or about the QT mutation.
Yeah.
I think it's a very misleading paper and very dangerous, because it gives the illusion that someone, even the best cardiologist on earth, can predict the effect of a non-synonymous mutation on a patient, which is absolutely untrue. It's impossible to do that now if you see what is a mutation, and you have to do very complex biology. Now, the important thing here is not do you have a mutation, but do you have your father who had the same mutation who died, or you have a sister or brother, or is there someone in the world who had the same mutation, who had the same disease? That's the only interesting thing. The other thing is literature and research. So I think we have to be very, very careful by sequencing normal people and saying you will die. No, probably you won't. But
I take your point, but on the other hand, you just argued that everybody should be typed on some kind of chip so that they have all their variant information. So you can't have your cake and eat it too. The problem with the JAMA paper was that we did a bunch of middle-aged people who didn't have phenotypes, who were survivors by definition. And so the likelihood, this is all about Bayes' theorem, the likelihood that those people have the congenital long QT syndrome is small, and that's the pre-test probability. The post-test probability doesn't go up a lot. On the other hand, if you have people who have a strong family history of sudden death, and they're in my clinic because they have a genetic arrhythmia problem or their QT interval is long, then the pre-test probability is high and the post-test probability is equally high. So I think that's the problem. But I think you're highlighting the biggest issue, and that is we find people who by rights should have a phenotype and don't. And that's what happens when you start to go into the population. And so I think that as we go into the population, we're going to have to be very, very sensitive to this idea that what we thought of as highly pathogenic variants may not be in every single population. I'm sorry to have interrupted, but
precision medicine is very much about DNA, and I think with our ability to analyze DNA, we are finding some very astonishing things. So a couple of years ago, we found all these bits and pieces of DNA in human blood, which were not coming from the human genome, but from bacteria. Yeah. Which led us to the microbiome and the bacteria in the gut. I'm wondering, my question is, how will the US Precision Medicine Initiative will be able to adapt to the evolving science of the microbiome?
Well, I think so you're right that there's this way that people have of using words that equate precision medicine and genomic medicine in the same breath. And precision medicine is a much broader concept because it takes into account not just the genome and the metabolome and the transcriptome and the microbiome perhaps, depending on logistics, but also things like how far away from a grocery store people live, what their income is, what their educational level is, do they understand, they have 6 medications, do they understand when they're supposed to take which, do they understand when they're supposed to go to Coumadin Clinic to get their INR checked? All those other determinants of health that are probably more important than the genome in many, many instances. Whether the Precision Medicine Initiative will be able to accommodate the microbiome, I leave to the people who are running the Precision Medicine Initiative. There's a lot of excitement about collecting not just DNA, but maybe even cellular material, maybe other kinds of material, but the project is in its early planning stages, and the question of the microbiome has been raised before, and I'm just not sure what the answer is going to be to that. I'll just leave it at that. The one thing is that the cohort that's being built is recontactable. So one thought is that if you find people with interesting phenotypes, then you go back and say, in these 20,000 people, we'd like microbiome samples of some kind. So that's going to be on the table, which our resource at Vanderbilt, for example, is not recontactable. So we couldn't do that, but they can. And that'll be a big advantage.
One last question.
Thanks very much, Dan. This beautiful discussion. My question really follows a little bit on the last question, is in a hotbed like Vanderbilt, where you've been focused on pharmacogenomics, what have been your thoughts about the other omics in terms of the molecular phenome? Maybe coming back into the host rather than the microbe. If you think about the proteome, the metabolome, and the transcriptome, and regulatory or non-regulatory transcriptome, to get that either the extreme phenome or going in that direction or coming the other way from the genome, what are you folks doing at Vanderbilt in terms of going forward with bringing it all together?
So the biggest, it's clear that there are some phenotypes that where variability lives in the genome, and there are many, many, many other phenotypes, probably the vast majority of human phenotypes, where the variability lives in the genome, but in other places that you could measure. The long QT example that I gave is very parochial, but it speaks to the idea that what you need is actual source material from patients who have known phenotypes. You're going from phenotype back to, I won't say genotype, because what we're going to do with these cells is we're going to look at the end result, the electrophysiology, we're going to do RNA profiling, we're going to do gene sequencing, obviously, but it's a matter of looking at multiple data sets, multiple overlapping data sets, to come up with an answer that you believe. And so in some instances the genome is sufficient. In other instances the genome is not only insufficient but probably doesn't give you an answer that was worth doing. Another project that we're doing that's very similar to the drug-induced arrhythmia project is a Stevens-Johnson syndrome project, where we're actually trying to get patients who have this terrible drug reaction while they're hot and get the blister fluid from the Stevens-Johnson syndrome patients, because we want to get the T-cell populations in those blister fluids. So that's something that a DNA bank cannot possibly do for you. You have to go back to the individual subject. And I think that's where some of precision medicine is going to go back to, sort of in exquisite phenotyping at the molecular level of individual subjects with known phenotypes, known clinical phenotypes.
And just a footnote on that, I think in terms of that global network of biobanks, one of the challenges, of course, is the cost and logistics around the right biospecimens with the right SOPs in order to go back there someday and bring it all together. But thanks.
I agree completely.
So thank you again, Dr. Roden, for this very productive discussion we had after your talk.